Hello, my name is Kayla Wallace, and for my senior thesis, I explored the effects of freshwater acidification and parasitism upon rusty crayfish consumption behaviors. Aquatic ecosystems are a vital part of the global environment. However, the security and stability of these systems is threatened by climate change. The Great Lakes region is one of the largest freshwater systems in the world, providing invaluable services, jobs, supporting human and animal life, both within and beyond the region. However, this region is threatened by climate change in the form of acidification. This poses unique risks to acid-sensitive organisms like the rusty crayfish, which is a keystone species and ecosystem engineer for the region. It does this through compromising their physiologies and biological processes. However, the stress of acidification will not occur in isolation, but rather alongside biotic stresses like disease and parasitism. This study seeks to investigate the individual and interactive influence of acidification and parasitism upon crusty crayfish with my 3 questions. First, how does pH influence rusty crayfish consumption? Second, does parasitism induce self-medication in crayfish? And third, what is the combined effect of parasitism and pH on crayfish consumption? For my experimental design, I subjected 40 female rusty crayfish that had varying degrees of parasitism to a range of pH treatments and conducted foraging trials with them after they habituated to these conditions. I measured their consumption of macrophytes, and I selected 2 macrophytes. The first, pictured in figure 1, Potamogeton-Richardsonii, which has medicinal compounds that enable an immune response to kill off parasites, and thus would be an index of self-medication. And I also chose the species Cara, which is a normal staple in the crayfish diet. After completion of the trials, I dissected each crayfish and photo processed it using NIH ImageJ software through the steps pictured below, and essentially quantified immune response and parasite load through that photo processing. To investigate drivers of overall consumption and preference for P. richardsonii, I fit linear effects models using GLS for overall consumption, which is on the left, and using LME for the P. richardsonii preference models on the left. And I chose the top models based on AIC values. Through analyzing the top models, I observed many trends. For overall consumption, I noticed that pH and parasite load were frequently grouped together in the model output table. So I divided pH and parasite load into one class and length and location into another, as length and location were also frequently grouped together. And you can see that pH has a positive effect on consumption, meaning at higher pHs, crayfish consume more, and at lower pHs, our acidic conditions, they'll consume less. Parasite load reduces the amount that they consume, and the interaction between pH and parasite load overall increases the amount that they consume. Length and location had larger effects on consumption, with location being the greatest with an effect size of 1.40. And it was interesting to look at how the locations compared. For crayfish from Burt, they had a significantly higher parasite load of around 5.7%, while Munger had a lot lower parasite loads. To summarize these results of my overall consumption, location largely shapes consumption. Parasite load varies greatly by location. Crayfish eat more at higher pH and less at lower pH. Parasite load reduces consumption. And lastly, pH and parasite load interact to have a positive effect on consumption. The P. richardsonii preference models were very interesting as well in the findings that they revealed. Essentially, I saw that parasite load increases the effect of P. richardsonii consumption, as does melanization, which is their immune response. And all of this points to self-medication. It is very interesting, however, that between location, the P. richardsonii consumption varied. You can see that for Burt crayfish, it was significantly higher than for Munger crayfish. So there's something between the locations that is leading in a differential preference for P. richardsonii and resulting in Burt crayfish to self-medicate, but not Munger. So to summarize, crayfish show support for self-medicating behavior and the percentage of P. richardsoni in diet varies between locations. To conclude, I found that location largely shapes consumption of the high phenolic or medicinal plant P. richardsoni, as crayfish from Burt consumed a lot more P. richardsoni as compared to Munger Pond crayfish. But the 23-fold difference in parasite load between locations is worth noting, as high parasite load is likely the cause of the self-medication that we see in Burt crayfish. And I'd like to reemphasize that this study supports the presence of self-medicating behavior in crayfish. This is seen through the way that P. richardsoni consumption increases with parasite load and immune response, and the positive effect that immune response has alongside P. richardsoni consumption, thus suggesting that it is acting as a medicine, enabling parasites to overcome parasitic, or crayfish to overcome parasitic stress. The consumption of crayfish is jointly shaped by biotic and abiotic factors, however, and climate change will result in acidic Great Lakes, presenting a new stress for aquatic life in the region. From the positive relationship we see between pH and consumption, this tells us that projected acidification is more likely to result in less macrophyte consumption by crayfish. The interaction between parasite load and pH increases consumption, which gives insight into how crayfish may respond in the face of these dual stresses. However, it is difficult to disentangle what this interaction means at lower pH conditions. The interaction, in addition to the interaction between other changes to the freshwater climate and parasitism, should be examined in the future. However, this study does reveal a fascinating insight that pH and parasitism must be considered together for a comprehensive understanding of the future of the rusty crayfish populations in the Great Lakes region. I would like to acknowledge Trey Scott and Joan Strassman as my thesis mentors throughout this process, and furthermore, acknowledge Trey Scott again for his patient assistance in model generation and analysis. I'd also like to acknowledge the University of Michigan Biological Station, Paul Moore, Becky Mackay, the Moore Lab, my RU cohort, Stephen Bertman and David Caraway for their support throughout collecting the data at the University of Michigan Biological Station. Lastly, I'd like to acknowledge funding from the National Science Foundation and Department of Defense for funding this project. Thank you.